Hello everyone. Now as we have already discussed the PCR technique in our previous video, so today we are going to learn a new technique which is called RAPD or Random Amplified Polymorphic DNA. It is a PCR based technique and we use them to identify the genetic polymorphism. Now polymorphism means there are variation in the gene structure among the organism. So if I want to identify those polymorphism among various organisms or among various individuals within the species, then we can use this technique which is called random amplified polymorphic DNA. Now this is a PCR based technique, but unlike PCR reaction, here we don't need any information regarding the sequence we want to amplify. Because in the PCR we have to design the primer, so that's why we should have a little bit information about the DNA which we are want to amplify. But here we don't need any prior information or sequence information of the DNA strand. Here we actually use only a single primer. In case of PCR, we generally use two primer. One is the forward primer, another one is the reverse primer. But here in the RAPD technique, we actually use only a single primer, and that single primer is very short, and that particular primer, the single primer, can act as the forward primer as well as the reverse primer. Because the size of the primer is comparatively very low in case of RAPD reaction, where the primer size generally varies from 8 to 10 nucleotide in size. And if there are any palindromic sequence present within the genome, then the same primer, they can bind in the palindromic region, and the single primer, they can act as a forward primer as well as the reverse primer. And that's why, which ultimately leads to the amplification of a stretch of DNA. Suppose this is a DNA sequence, right? Manu, this is the DNA strand or the genomic DNA, and Manu, we have designed a primer which is 8 nucleotide long. Now Manu, the primer sequence is complementary in that particular region. So the primer will bind here, and Manu, the same region, they have a palindromic sequence here. If they have the palindromic sequence here, the same primer will also bind here. Now the same primer, they can act as a forward primer as well as a reverse primer. As a result, this primer will amplify this segment of the DNA after the PCR reaction. So we are going to use this particular technique in the RAPD. Now what we have to do in the RAPD reaction? So during the RAPD, first we have to isolate the genomic DNA from the organism we want to compare. Manu, for simplicity, we are just going to compare two organisms, organism A and the organism B. Now they can be come from different species or different genus or different family. So, and we can also compare more than two organisms at the same time. But for the simplicity, now just consider we have two organisms, organism A and organism B. Now in the first step, we have to isolate the genomic DNA from organism A, and we also have to isolate the genomic DNA from organism B. And suppose these are my genomic DNA. These are my genomic DNA isolated from organism A and organism B. Now we use this genomic DNA as a template, and we will use an RAPD primer for the RAPD amplification or for the PCR reaction. And suppose in case of organism A, in that particular genomic DNA, the same primer, it has the capacity to bind here, here and here, because these are the palindromic sequence. And as a result, when you perform the PCR reaction, the primer will bind here and here as they are the palindromic sequence, and it will give an amplified product, right? And the same primer will bind here and here as well, and it will give you another amplified product. Now when we analyze this amplified product in an agarose gel electrophoresis, so this is the agarose gel. So when we perform the agarose gel electrophoresis, these two DNA fragment or these two fragment, they will give two band. But here we don't have any previous information about the DNA. So the DNA which are going to amplify is at random, because the primer will bind randomly. Whenever it finds a palindromic sequence, it will bind there and it will try to give you an amplified product. So as the amplified product are completely random, that's why you call it random amplified polymorphic DNA. So in case of organism 1, so these are the 4 binding sites for that particular primer, which ultimately leads to the production of 2 separate DNA fragments. Now as we can see, this is the smaller fragment and this fragment is a little bit larger in size. So when we perform the agarose gel electrophoresis, the smaller DNA, it will give a band here, and the larger DNA, it will give a band here. Now comes to the organism B. Suppose during the evolution in the organism B, there was a deletion event. Because we are talking about the polymorphic DNA, and the polymorphic DNA, they can arise due to the mutation, or there are any changes in the genome, that can be insertion of a particular sequence, deletion of a particular sequence, or point mutation in a particular sequence, which can give the help in the generation of the polymorphic DNA, or it's actually bring the genetic variation in the society. So Manu, in this particular organism there was a deletion event. Now due to that deletion event, Manu, this portion, suppose a small fragment has been deleted in organism B. As a result, the primer, it will not able to bind in that particular area, because this particular position is actually missing in organism B due to some deletion event. Then what will happen? Now the primer, the same primer in case of organism B, it will able to bind here, here and here. So as a result, these 2 sites, they remain intact. So it will give you the amplified product, which is just equivalent to the larger fragment present in the organism A, but the smaller fragment is missing, because the second site where the primer should bind to give this amplified product is missing due to a deletion event. Now when we amplify the product and we run in an agarose gel, it will give you 1 band. So now this profile will indicate that there is a polymorphism among these 2 organisms. That's why we can analyze this polymorphism by just analyzing the agarose gel. Now in reality, when we actually perform the RAPD in a molecular biology lab, it's not only give you 2 bands or 1 band, because when we use a short primer around 8 to 10 nucleotide long, they at least give you near about 100 bands. So what we have to do, suppose we isolate the genomic DNA from multiple organisms, starting from organism A, B, C, D, E, F, and then we have to isolate the genomic DNA from all the organisms, and then we have to perform the PCR analysis. And for every individual, it will generate a banding pattern. Here, this is the banding pattern, this is the simplified banding pattern for organism A and organism B. But when we actually perform the RAPD reaction in a laboratory, so Manu, for one organism, Manu, for organism A, it will give you hundreds of bands, and it will give you an unique profile for that particular primer. Now, for the same primer, for organism B, it will give you a separate type of banding pattern. And Manu, for organism C, again it will give you some unique type of banding pattern. So this banding pattern will help us to compare between the polymorphism between this individual A, B and C. And not only that, it will also help you to identify the evolutionary pattern. Okay, the evolutionary similarities between these 3 organisms. Mano, in A, B and C, when we have the RAPD banding pattern, we can compare the banding pattern and we can analyze that which one is a close relative. Mano, among A, B and C, we want to identify whether B or C, who are the close relative of organism A. So in that case, the banding pattern of B and C, we have to compare them with the banding pattern of the A. The individual which will exhibit the closest banding pattern of organism A, we can say that these 2 individuals, they are closely related, and the individual which will give the diverse type of banding pattern with individual A, we can say that particular individuals, they are distantly related with that particular organism. So this is the molecular basis and how we are able to perform the RAPD, and now we are going to discuss the application of RAPD in real life. Now we are going to discuss the application of the RAPD, because till now we have discussed the molecular basis and how we are able to perform the RAPD. Now we are going to discuss the application of the RAPD technique. The first application is in the genetic mapping, because RAPD is basically a molecular marker. There are some other molecular marker as well, like RFLP and AFLP, which we are going to discuss in our next videos. Now today we are only discussing the RAPD. It's a molecular marker, and that molecular marker we can use to map the genome of an organism. So this is the prime application of RAPD in the genetic mapping. Now along with this particular application, there are some other application where we can also use the RAPD technique. For example, identification of the varieties, because as we already discussed that the banding profile or the RAPD banding profile can be used to identify the relativeness between multiple organisms, which we can use to identify the multiple varieties of a particular organism. Along with that, we can also use the RAPD technique to differentiate between the strains of the pathogen, because when you're talking about the different strains of the pathogen, they are very closely associated, but there are little bit modification and changes in their genome, and that's why they give rise to different strains of a particular pathogen. Now in that case, we can use the RAPD technique and we can analyze the banding pattern, and that banding pattern help us to identify the strain of that particular pathogen, because some strain of a pathogen they may be virulent, some strain of a pathogen they can be avirulent. So the identification of strain is very important, and that identification we can perform using or taking the advantage of the RAPD. And next is the molecular phylogeny, because using the RAPD technique we are able to analyze whether the organism they are closely related or the organism they are distantly related, which help us to as a evidence to help to construct a molecular phylogeny. Because the in case of molecular phylogeny, we actually try to draw an evolutionary tree using the molecular data or using the DNA sequence. So in that case, when we propose a phylogenetic model, that RAPD technique can also help us to reinforce the data that whether the predicted phylogenetic tree is perfect or not. This data can also be analyzed using the RAPD technique. So that's all for the RAPD. Thank you.